So Larry Boyce is a professor and vice chair of basic research at Emory and also at the Winship Cancer Institute. And you have let me just grab everybody else in here. Aya Khalil is the chief commercial officer and executive vice president and co-founder of GNS Healthcare. And Kate Sasser is the senior director of hematology oncology translational research at the leader at Janssen R&D. Part of Johnson & Johnson. So this is kind of an all-star crowd, and I want to just tell you a little bit about each person before we even get to the questions here. But so Larry is just sort of known as the sort of heavy hitter or one of the heavy hitters in myeloma research. You actually have a title, you're Georgia Cancer Control. What's your title at your professor?
The Georgia Cancer Coalition distinguished, cancer scientist. I can't remember anymore.
Because all the chairs when I went to school, it was like the Alfred Lord Whitehead Professor of Punishing English Students and stuff like that. It was never like that. And Aya has started a couple of companies. She was just honored by the President of the United States in a private dinner as being one of the leaders in genomic science, and she was just named to one of the Pharma Voice 100 for building bridges across lines. And then don't look at the YouTube video because it mispronounces Aya's name, but we won't talk about that. And then Kate is, of course, one of the team leaders on daratumab I probably mispronounced that, but at Janssen, close enough. So monoclonal antibody, that's all I need to know. That has been really on the forefront of looking at this new targeted myeloma medicines and a real leader in the community. So it's a great group, and we're going to talk a little bit about moving from say this open access data to actual research, to what we actually can do with all this data. So the first thing I want to throw out to you, and let me start with you, Aya, in the middle, because you actually manage all of these giant data systems, and you happen to be a theoretical physicist. So you were describing something earlier about a problem that Aya had worked on in studying theoretical physics, which is the many bodies problem, which is that you have two bodies right next to each other, say an electron and a proton, and they sort of manage to have some sort of coexistence, and there'd be some movements and forces will work on them. But the minute you add another body into it, everything goes a little crazy, right? New forces come on. So talk a little bit about what happens with biology and why this data is so important to you.
Yeah, thank you. Thanks for bringing up the physics analogy. So President Obama said I was a recovering physicist. So I still think I am, so I apologize for that, but that's how I see the world. So as you had mentioned, in physics, the minute you introduce a third body to a system, it automatically becomes complex. And complex in that you have all sorts of emergent behavior. And here we have biology where we're dealing with infinite amount of complexity once you consider that it's not just three kinds of particles, but many, many kinds. We're dealing with our proteins, membranes, cells, our environment. Everything influences the system, and it's a hugely complex system. And so what's been great about working with multiple myeloma is when we think about complexity, we first think about how do we first measure it. And what we've done here is gone through and taken a cohort of patients and actually measured lots of data modalities that help us first capture what we're seeing, what the system is telling us, and at multiple levels. So it is your genome, it is molecular expression, the things that go in, the workhorses in the cells, but it's also your phenome, and this is really important, that we're going from the very granular to phenotypic data to clinical data and capturing measures that we hadn't been able to. If I go to an EMR record and I want to go in and see who's been newly diagnosed, who had some kind of first treatment response, I'm not going to be able to find it. So we're capturing now the data, many, many different levels, and we're doing it longitudinally. So we can see what happens at baseline when the patient presents, but also as the disease progresses. So this is great. We have this data, and the data is now helping us get a window on that complexity and what it looks like for this disease. But the second level is, what do we do with that data? How do we analyze it to get insights? So I'm going to throw one more physics analogy, and then I'm done with physics, I promise you. But it's around, so we recently discovered the Higgs boson. And that was a collaboration to actually get to that discovery confirmation. Many, many different sciences building huge machines, lots of complex technology. The analogy of what we're doing here is really similar. Lots of complex technology. We have to measure things using genomic sequencing, many, many different scientists. But also the taking in of that data now and generating algorithms and math that can explain the mechanism underlying that data so that we can make this prediction and verify it. And if we can do this to make a prediction around the God particle, then hopefully we can now bring this back to patients and make predictions about what the right treatment is for the right patient at the right time. So when I think about the algorithms and the math that we need to deal with this complexity, it has to go beyond just looking at patterns, right? Looking at correlations, because in medicine, patterns and correlations will mislead you. Leads to all sorts of confounding predictions that don't pan out. We need to be able to predict what happens if I make a change to the system. What happens if I make an intervention? Will you respond better or worse? And it turns out that there's an underlying set of mathematics that's been developed to help us learn causality in a probabilistic way, because it's still medicine, it's not deterministic. And we can start to tease out and learn causally what's happening. So when we talk about learning about the system and the disease in patients, we're talking about learning the networks, the complex networks that link your genetics to your molecular profile, to phenotypic measures, to clinical measures. And it's a network. And once we learn that network, we can start to make accurate predictions about what happens if I intervene and get down to a very precise prediction for that patient.
Now, we hear a lot about big data,
Yeah.
but with the COMPASS study, it's not just big, it's deep,
Yeah.
right? So we've got whole genome sequencing at multiple stages of patients. We've got not only 1,000 plus patients that we're working on getting, but it's also RNA, transcripts, it's a whole mess of other stuff. Larry, what do you look for in the lab, or what are you looking for now, and what's it telling you, this data?
So, thanks for the question. And actually, before I start, just thanks. I'm so happy to be here, actually. Thank you for the invitation, and Paul and Kathy. The MMRF actually got me into doing myeloma research. I was just playing around with it as a hobby 15, 16 years ago, and it was actually funding from the MMRF back then that really solidified our research efforts there, and they maintained them there.
They get everybody into MMRF. There's a diner down the street, and they're all doing myeloma research.
And now what the MMRF has done is it's really changed the way we think about these research questions. So I'm not a physicist, I'm a biologist, and we still think about biological approaches to understanding a disease. But now we don't ask a question. All our questions start with COMPASS. And so we think about, okay, we're interested in a pathway or genes that are involved, that may be involved in controlling a pathway. My favorite pathways happen to be ones that decide whether a cell lives or dies. And those are important because those are how our therapeutic approaches end up ultimately killing the myeloma cells or not killing the myeloma cells, as it were. And so that's where my basic science interest came from. So now what we do is we just go to COMPASS. So we have a question now. The first place we go is COMPASS to say, well, is this gene that we're thinking about, is it relevant in myeloma? So we can look at the clinical data and say, who are because of the transcriptomic data, do the patients who have high expression or low expression of this gene, do they do differently? Are there mutations in the gene? Because there's now 900 patients sequenced. And we can look at that and say, well, are there mutations? And if there are mutations, how do those patients do? And so we start all our questions now with COMPASS and looking and using all that data, whether it's the clinical data, the genomic data, to build the questions. And then we ask biological questions. And so one example recently is looking at one of these genes that actually regulates cell survival. We actually looked through about a dozen of them that regulate this, and it turned out there were only mutations in one single gene in more than one patient. And it's still only about 1 or 2% of patients that had a mutation. But all the other genes, there was only maybe one patient had a mutation. So, but this one had there was a bunch of patients, 1 or 2%. And so we went through and we then modeled what those mutations would do to the structure of the protein that's made by that gene. And we modeled that and said, okay, that's interesting based on what the clinical data was, because the clinical data was those patients were doing better. And now we think we know why. We think that the mutations are actually causing the protein not to work quite as well, which would make the cells more susceptible to therapy. But that doesn't prove it either, so that's still correlation. And so in a biological approach to looking at this, what we'll do now is we're going through and actually testing this in cells. So we're making all of these mutations in the gene and then testing it in biological systems to see if it actually acts exactly the way we would, which would then fit with the clinical data. And so it now focuses all our questions. Sometimes it doesn't focus me though, because the things like the Researcher Gateway and the GNS healthcare system, they become my video games, right? So if I have anything, instead of playing, Candy Crush or Pokemon Go like my kids are doing, I'm on these programs and I just have a question and I just start going away and start trying to find these. And so it's generating. It's not just how we start our answers. It's where we're starting all our questions these days. And that's really what having such a deep database allows us to do.
I love that. Kate, so you're in the Dream Factory. You're in the place that makes medicines, I like to think of it. But there's a misconception. So when people out there think, oh, all we need is a target for a drug, well, you've got a good target. You've got CD38, which is expressed on multiple myeloma cells. But tell us about the challenge of finding out why say a drug doesn't necessarily end the disease, even though you've got your target and it homes in on it, and what you can learn from this rich database to help you sort of find the way.
Yeah, great question. And I love the Dream Factory.
Yeah.
I'm going to go home and tell the team that, because I do think that, data sets like this really help us in many different ways. And I think, we've talked so much about data and having large data and lots of data and adding to that. But if you can't translate it into improvement in the field, improvement for patients' lives, new therapies, a deeper understanding of the therapies that we have, then data by itself is not that useful. And I think that's why we're all here and why we do what we do and why we're scientists to begin with. So you're right. At the beginning, I remember somebody had a table that showed all of the therapies now that are out there for myeloma, which is great improvement in the field. But the truth is that even with every single one of those, and even with new drugs like the monoclonal antibodies and daratumumab that you'd mentioned, we still have patients you came close. It's good. We still have patients that ultimately either don't respond or they respond for a while and then they relapse. And that tells you, as we heard also, is that we're dealing with really complex systems. When we start to get into drugs like the antibodies, you're talking about a target on the surface of the cell. So now we have to add in the immune system, and this particular drug works by attracting other immune cells to that myeloma cell so that they can actually kill the myeloma cell. So now we have to think about the patient's immune system and those effector cells, as it would be, not just the tumor cells. So now we have to add in these other complex features. And so what we can do is we can go back to data sets like COMPASS, where they've measured not only the genomics of the tumor, but they've also pulled in surface profiling of the myeloma cells, so we can look at the surface phenotype. We can start to look at the immune system. So we heard earlier about how they've actually collected samples to be able to profile the patient's immune system, and to do that all along the course of therapy. So we start to learn, maybe at a newly diagnosed stage, there's a certain immune phenotype that goes with certain genetics. And then maybe as patients relapse, whether it's an IMiD or a proteasome inhibitor, or in a few more years it'll be the monoclonal antibodies, a few years after that it'll be T-cell therapies. As we start to gather that data, we can really start to, with the help of Aya and her team and others like them, be able to put it together and look at this system as an entire complex organism that it is. So it's not a single mutation, it's not a single phenotype. It's really about all of the data together. And I think that's where having the data set, ultimately our goal is to be able to do that analytics faster, to be able to break things down quicker, to find new therapies, and to improve on the ones that we have. So.
Okay, in exchange for using the Dream Factory, give us a trade secret. Tell us something you actually learned from the COMPASS data.
Turn the video off.
Turn the video off. This is why you pay the big bucks to come here.
Right.
It's right here.
Absolutely.
Well, I will tell you that one of the things we have learned from the COMPASS data set is when we have our scientists at the bench who come, running into the office and, oh, we have this great new target. We've taken a whole panel of cell lines that have been in the lab for years and years and years, and they did some new experiment, and they think they found something, maybe a new gene or maybe a new surface protein, and they come running to us and say, we really should move this forward in the pipeline. And we go to our board and we say, okay, well, we've got 20 of these things. Which one are we going to fund and take forward? One of the great things about Compass is we can go back to that real world patient data set and we can ask the question there. So we can say, okay, we found this thing in the lab in these mice and it looks pretty good in the mice, but we're not quite sure what it looks like in the real patient setting. What did it look like in the Compass data set? And that will give us some insight as to how that might play out in the real world. And that adds some validity. It helps us make decisions faster within the dream factory world, where a lot of times it comes down to funding for those projects. So there's a lot of crunch for funding, obviously, in the academic world. We face some of the same crunch for funding on the pharma industry side. So we still have to make decisions about what moves forward. And data sets like that help us do it quicker and hopefully make better decisions as well.
So so there's a famous progression that goes data to knowledge to data to information to knowledge to wisdom. And to get from data to knowledge, it takes a lot to sort of compile a lot of data to knowledge. From knowledge to actual from data to information takes a lot. From information to knowledge takes a lot. From knowledge to wisdom takes a lot. Tell us what pieces of data you would need, what sort of combination of data you would need to really get something that you would consider truly informational, and what would be the next step to actually give you something where you say, wow, I can take this information and act on it.
Yeah. So I think the key thing is that the integration of different data types, really, really important. So we're going in and we are looking at genetic variation, but that's still not enough because genetics, not deterministics. Look at molecular variation, okay, but how does that then influence phenotype, different clinical endpoints? So integration, multimodal data is key to these big data efforts being really successful. We can look back, and this is no joke, we can look back historically at money spent on GWAS efforts where people didn't do a good job of clinically annotating those data sets. And so I will say something controversial.
Can you explain what that means for everybody?
Yeah.
When you say clinically annotating, what are we talking about here?
So what is the clinical profile of patient? Did they respond? Did they not respond?
To a specific treatment.
To a specific treatment, characterizing response accurately. So if you go into an electronic medical record and you try to figure out who the responders, non-responders are properly diagnosed, I mean, you're not going to. It's not properly captured in these records. Right. And it's that's one of the gaps that we need to bridge in medicine, because you would like your EMR record to capture all of these things really well. What we've done here at the MMRF is said, look, we're going to get this, so let's work with the physicians, with the patients, so that we can get the right clinical measures, get it down to the quality that you would get from a clinical trial, which is a lot of what pharma companies have to do to prove their efficacy of their drugs. Let's capture those same measures in the real world. So now we have that data that's multimodal, that integrates across different levels of information. Then it's the algorithm that says, how do the combination of factors together predict outcomes and predict the networks, the biological networks that drive patient outcomes. And now we have the models. And you said something about wisdom, and I think this is really important, and both of you touched upon this. So these models, they should not be in the hands of physicists. They need to be in the hands of the clinician, of the biologist. As you said, I love what you said about video games. Like, now I'm going to go in, I'm going to ask my what if question. Right. I just saw a patient and I saw something interesting about their biology. Well, what if I go in and I upregulate this, downregulate that? Will I see a better outcome? Oh, and by the way, I have an idea of a drug I could reposition where I could make that happen. So that community of scientists that can now look at data in a different way, data at the level of associations and interactions, and play with models that allow them to ask the what if, and then they go in and implement it, and is the way we get to wisdom, in my mind.
Larry, so you're used to collaborating with other academics, other scientists. You go to cancer meetings, and you share your data, and I believe that there's a fair amount of collaboration on that front. How has the data from Compass and this sort of convening power of MMRF allowed you to collaborate outside of that academic, say, silo, if you will?
So most of our, so one of it would be through the MMRF, and we've had these great data jamborees actually associated with Compass over the last several years that Daniel has actually
Like Burning Man, right? It's just like you all.
Exactly.
Okay. I'm picturing it now. I'm just trying to, okay, gotcha.
It is. It's actually, I believe you guys actually hosted one of them as well. So we did, and it's taking people who are involved in Compass from all different aspects of whether it's GNS, from people from industry, people from academia, actually a lot of the people in the room who were there at the last one as well, and getting into a room and sitting there and people presenting data and ideas on how to work on them, and even breaking out into small groups and thinking about how best to use the data and how to move forward as a team. And I think that's been one of the best ways that we've done this. We're moving outside of just academia. Mm-hmm. Within academia, for me, one example has also been a colleague of mine who's a structural biologist. He doesn't really think about disease at all. He thinks about protein structures, and he does an X-ray crystallographer, and he made a structure of a molecule called MAX. That he was working on. And MAX, it turns out from the 200 patient data set, was one of the ones that came up as mutated in myeloma. So he had all this data and he was looking at how MAX was working, and he even made some of the mutations. And so when I heard about this, we went to Compass, looked it up. Sure enough, some of the mutations he was modeling doing structural biology were mutations that were from myeloma patients. And so we immediately added a whole section into his paper using Compass data on the survival of the patients who had these mutations to help explain why those mutations were affecting the biology. So it's not only just going from academia, industry, biotech, outside of there, but even within academia, bringing people in who would have never thought about, who probably didn't even know what myeloma was really, but they were doing really interesting basics, structural biology, and then bringing that into our knowledge base and understanding the disease. And for him, helping actually making the data much more relevant to what they're.
I've traveled the cancer world for some time. I've never come across a data set this sort of well characterized, this deep, this sort of broad in its scope, the questions. Does anyone know of anything in another disease setting where there would be this kind of study?
Yeah, there's been outside of cancer a couple of examples to note, and it's actually, they all have the same features, which is the care it takes to ensure that you're capturing data on the different levels, be it genetic, molecular, phenotypic, clinical. So another data set that we're working with is in cardiovascular disease with an entity called Global Genomics, where they did something similar, multimodal data at different levels. And it's just generating insights that you would just never think possible. We're relearning the pathways around LDL biology, this is what we know. And this is part of the surprising thing, is we're looking at the models and we're seeing the things that we know in a different light completely. And then new things, wait a minute, there's this new player and pathway that's relevant for the disease. No one's looked at it yet. Mm-hmm.
So do you want to take that too? Yeah.
I think it's not only, I'm in cancer, I'm not aware of this. I mean, there were all the TCGA projects, but again, as we've heard, we're bigger, and much deeper than any of those projects that were done. So and it's such a rich data set for that reason. But I think for me, the most important thing is I'm a trained biologist, as I said. I'm a biologist because math, I wasn't so good. Unfortunately, I had to go back and relearn math because of that's what biology has become. But I went into biology. That's what I was good at. What they've done with Compass, thanks to people like Jonathan, is made it actually something that biologists or clinicians can actually use. So you don't have to be a bioinformatician or a mathematician to gain immediate access to the data and usable data. Being a bioinformatician or a physicist or mathematician, that helps. I'm sure you can do much more advanced things, but anybody can use it. So it's just easy to use, rich data set. I don't think that exists anywhere. Mm.
And Kate, coming from the industry perspective, what's extraordinary is the access that industry has to this as well, and can contribute to it too. Mm-hmm. Can you talk about what the sort of give back is in terms of new reflections on the data and sort of new science that gets added to this?
Yeah, well, I mentioned speed of development, which I think is a big one, and we take that very seriously. So we're moving things faster and faster into the clinic so that we can hopefully continue to make a difference. So we're not stopping just when you have one drug that looks like it's going to be a really good one. We're continuing to move others forward.
In the Dream Factory, yeah.
In the Dream Factory, yes, with the Dream Team,
Yes,
hopefully.
yeah.
The other thing I would add to that is that we see this as really being not just about new therapies. So there's other aspects to patient treatment that we think are also really important and that are part of COMPASS. So I'll give you one example. We partnered early with the COMPASS study because we wanted to look at other tests that could be useful for patient care. This particular example, we were interested in getting away from bone marrow aspirates and starting to move towards something that was whole blood so that patients would not have to continually have bone marrow aspirates all throughout the course of treatment. And so, at very early stages, we sort of came to the MMRF and said, we have this prototype test, we'd like to be able to include it and start to gather data and see if this could really work. And lo and behold, that's been an incredible partnership. So that test was incorporated into COMPASS. We're now getting data back, and it's given us incredible insights about new diagnostic tools that we may be able to use in the future. Hopefully that improves patient care, improves our access to be able to do things like sequencing, but now instead of your bone marrow, it's in whole blood, in captured myeloma cells, and working with other researchers like Irene and others in the room. And so we see this as really being not just about new drugs, new therapies. I'll add one other thing that we're very interested in, and I think that when people think of industry and pharma, they typically think that we're really just focused on therapies that will work and then continuing to treat patients forever. And I can tell you that that's actually not one of our goals. So the other thing that we're very interested in that we've worked with MMRF on is, can we get to a certain point where patients no longer have to be treated? And how do we actually measure that? What does it mean biologically if you have a mutation that's at very, very, very low levels? And what level can we say, okay, you're good, at least for now, you can stop therapy, you can go out, live your life, hopefully have some less sleepless nights and come back and we'll test again. And so we're working on those type of things as well because we believe it's important.
I would love to get some audience reaction to this. I don't know how many people out there have experience with this, but just a show of hands, how many of you would prefer a blood test to a bone marrow aspirate? Oh, look at that. Look at that. Wow. So, those are the kinds of things that make a difference. I mean, they actually make a real difference. Obviously, stopping treatment is another thing with good effect would be a wonderful thing. One other thing, though, that I know that the data is, at least that's an area that you're studying, are there pre-malignant signatures? Is there a place within the sort of myeloma continuum or progression where you could possibly step in and interdict the issue so that we don't actually have to add the additional burden for patients of getting this terrible disease? Absolutely.
That's another huge focus for us. And again, we see this as being incredibly impactful to the community. So when we talk about data and how do we translate that, to me, you translate you want it to become impactful. So how do we make an impact if 5 years down the road with continued new therapies, we virtually can extend life for myeloma patients, then the new place to really make an impact, as we've heard a couple times from others, is thinking about being able to intercede far earlier and actually prevent people from ever getting myeloma. And we know, the I don't want to say good news, but the good news, at least biologically in this sense, is that we know for myeloma there is this pre-malignancy, this sort of smoldering phase, or even MGUS phase. So we're looking at that and being able to say there has to be an answer in there scientifically, once we have all the data and we have the analytics, to be able to determine if you're in that MGUS or smoldering phase, what kind of therapy you need and when you need it, and how do we attack it so that we can ultimately, 10 years from now, patients get smoldering, they get treated based on a therapy that works for them, and you don't have myeloma. That's really the goal, I think, of the community. And so, it takes all of us in this room doing that together. And we're focused on that as well. So.
You know, one of the beautiful things about the Compass study is that you can build on it, right? So you can add more information as you discovered, hey, we would really like to know about what sort of immunological markers we should be adding to this. Can you talk a little bit about that? I had just how we could incorporate new information, and what sort of information would you like? You've got a captive audience here.
Yeah, very good. So I think you've touched upon a couple of those. One is around getting profiles and data earlier, so that we can start to work on the prevention problem. And it's ripe now with the new immunotherapies. And as Kathy said, we can just imagine the possibilities. And if we can imagine the possibilities, then let's go and collect the data and actually make it happen. Other data modalities that better capture immune response, definitely high on my wish list. More longitudinal, we're starting to see, not that I'm happy to see them, but the cases of the relapse, we're going to learn from that. Why are patients relapsing? How can we prevent that and longitudinal progression? But I think the other, and this hasn't been brought up, and it's data that actually helps align incentives. And this is really important because everybody complains about this is so expensive. Who's going to pay for it? And so one important data modality is just what do these things cost? So we can get that from claims records, and we can incorporate that into our models, and we can exactly say what it costs us to treat a patient right and what it costs us to treat a patient wrong. It turns out no matter how much drugs you put on somebody, it actually costs more to treat a patient wrong. A paper just came out, precision medicine paper by the folks at Intermountain, that we know of and know of their work, where they have shown that you get better economic overall return if you do the right treatment, right time, right from the beginning. And why I say this is because I want to see these efforts continue to get funded and continue to get better, and for us to make the investments upfront in the data, because they do pay off. And not only do patients get better, but we save money. I mean, that's just tremendous.
I've got a bunch of great questions from the audience. So this is, can one's genomics accurately be analyzed after undergoing several treatments? Larry, you want to talk about that?
Yeah, sure. I think as long as there are cells to be analyzed, and I think we're getting better and better at having fewer and fewer cells required, but as long as we have cells to analyze, it can be done. Jonathan will do it.
Yeah. But there's a give and take. I mean, the disease may change on the basis of the treatment,
and I think
so you can figure that.
That's actually the most important reason, and that's actually what's real, and I think that was what Jonathan's point was about. The real strength of Compass is it's a longitudinal study, and we're going to be taking those samples at different points so we can watch those changes. I mean, he mentioned the clonal tide, and it was the one patient that got to watch it. It was one patient. It's a fascinating case. Let's see this in Compass. Let's see, how often does this occur? Is there any kind of correlation with changes that occur, even with relapse? What happens to the disease when it comes back? Can you do something different depending on how it's changed? So I agree. I think that's where the real benefit is going to be. It's going to be in years, 7 to 11. Is that what you said? That where we're going to be able to ask those types of questions, or answer those types of questions.
And Kate, this is a follow-up to your comment before. How close to the market is the new whole blood assay to replace bone marrow biopsy?
That's a great question. Who asked that?
There we go. There we go.
Nice.
I don't have a short answer for you. That's probably why you're answering the question. But I will say that data like what we're getting back from Compass helps us move it up, right? So the challenge with commercializing something like that, as you probably know based on your background, is really being able to show that it has viability as a product. And in order to do that, you need data to show where you can use it and how. And I think we've been able to at least make a really good case using Compass in our work with MMRF. So we think it has value in monitoring patients. You saw at the very beginning a slide where a patient who didn't respond well had very high circulating myeloma cells and the other one didn't. That's somewhat anecdotal because we were just sort of pulling those out. But you can imagine that if we see trends like that in the data, then we can start to put it forward as a commercial product. So that's the goal. That's what we intend to do.
So I think this was answered by your question, Larry, but I just want to make sure that, because somebody did put this in writing, but if you have no mutated gene at diagnosis, can a gene mutate after treatment? And if so, your genome has changed. So
absolutely, and the other possibility is, and thinking about myeloma as within a patient, it may not be a single disease even within a patient. There are multiple subclones of the disease, and it could be that in the initial diagnosis, the one clone was just that had the mutation was so low that it just wasn't really detected, and that after treatment on the relapse, that's what grew out, and so that's why the mutation is there. So it may have been there all along, but just in a small fraction of the cells, and that's what grew out. Alternatively, it could be a new mutation that comes out, following treatment. So either case is a possibility.
Can you all actually speak about the challenge of intratumoral heterogeneity? The fact that we've got different types of mutations in different, perhaps, cells in our own bodies.
I mean, so this is really, this is the next big challenge for us, right? Because we think about these precision medicine, and if you just use the analogy that I just said, where some of the cells might have had the mutation and some of them don't. Well, if you're treating based on the mutation, you're only going to hit the cells that have the mutation, and the ones that don't are going to be there. And so it's always going to be the challenge. And even in some of the original papers that came out from the MMRC, pointed this out, that it was somewhat sobering actually to see how much clonal heterogeneity was there, and that you could only, if you were going to go after a single target, it's probably only going to be effective if that target was clonal. In other words, it was in all the cell populations. And so where does that point to? It points back to where we're at in a way, and that we're probably going to still be using combination therapies. They just may be very different types of combinations, and they may be now more targeted to the patient based on the mutations that they have.
Kate, you want to address that too, specifically with your CD38 targeting mAb?
Yeah, I think this is a sort of fascinating, evolving space for us as scientists. So as we have these new therapies that I mentioned earlier, like daratumumab, that now work by targeting the surface of the cell and bringing in other immune cells to attack that tumor cell, we really start to add another question in the equation. So yes, the genomics of the tumor cell are incredibly heterogeneous. We know that's a problem across all of cancer, and certainly in myeloma as well. And as you treat a patient with therapies, it evolves, and so it gets more heterogeneous. You get additional mutations and changes. And now we have therapeutics that, on top of that, you may see cell surface changes. So it's not just the genomics, it's now what happens to the markers that are on the surface of the cell, or even with the immune system, right? So if patients have gone through certain therapies and have lower T cells or lower NK cells, how does that affect the therapy? So again, I would agree. I think it's going to be really interesting to think about combinations. Do certain combinations of a monoclonal antibody plus an IMiD, how does that compare to a PI? Or do you need all three to really get a big bang for your buck, so to speak? And this is really where the research is going. And it will continue to become more complex as we start to see things like T-cell therapies come into the marketplace. So, it's a fascinating world, and I think the bottom line is we really have a chance in myeloma as we start to have these large collaborative efforts to potentially figure it out, and it's a great case study to be able to show, this is what you can do in a type of cancer, and if we can do it there, we can do it other places.
Mm-hmm. Do you feel like that's addressed? Do you want to take a stab at it as well?
No, I think it is addressed, and it's hopefully going to be a combination. For those who have to worry about heterogeneity and noise, it keeps them up. How do you separate that signal from the noise? And so some of it is just algorithmic approaches that can help kind of understand what is the nature of the heterogeneity, what is noise, what isn't. But I agree, it's going to come down to combinations, and that's where unraveling those complex networks helps you figure out what are the multiple points you need to hit to do that. And if you can start to evolve that, get a longitudinal picture, then you can keep evolving that combination regimen.
So message noise, and this constant balance between what you think might be a message and it turns out to be noise, or trying to find the message in the noise. And Compass helps you sort of filter that out.
It does. I mean, again, and I think this goes back to some of the numbers issues that came up earlier. It gives us an opportunity to look at a larger group of patients, and that's to me is the way I think about, I think what Jonathan said about the statisticians being right, that's really what they're doing, right? It's filtering out the noise, and in this case, it's by increasing your number of patients that you're analyzing to look at these things.
Well, the value of open access, you want to tackle that? You want to just talk about what that really means to the scientific community and the patient community and the drug development community?
Yeah, I think it's huge. It's getting us all to a place where we can, kind of have common ground around the understanding of the biology and what drives the biology in patients. And we all need to be able to have access to that, whether it's on the data side, the algorithm side, insights derived from that. And then you create a framework where a pharma company is incentivized to create a new treatment, where scientists are incentivized to publish more and bring more insights. But these sort of siloed approaches to the data, oftentimes data they just get rusty after a while. Nobody looks at them, nobody gets insights from them, the community doesn't benefit.
Mm-hmm. What haven't I asked that I should have asked? Not answering. All right, here's a question from the audience. We've got 46 seconds left, and I've saved this one for last. It says, the title of your book about cancer is discouraging. Would you change the title now? So the chapter with Kathy Giusti in it and Paul, I think, is in like chapter 13. If I had begun that at maybe at chapter 1, maybe I would have changed the title to something more encouraging. Thank you all, my great panel. Larry, Aya, Kate, and thank you for your great questions.
All right, thank you, Clifton, Larry, Aya, Kate as well. Phenomenal. Thank you for taking these, brilliant PhDs and making what they said really digestible to the entire audience. So really well appreciated. I just wanted to emphasize, and then I'll let you guys all go to lunch, that this panel really represented that middle box for those 3 boxes that you saw a couple times in the morning, between the data bank where we're generating all this data and the clinic where we're bringing clinical trials to patients. This learning network where we're putting the data out there, it's being analyzed, it's being validated. What I loved hearing from this panel, and I hope it resonated with you as well, is the word speed kept coming up over and over and over again. And that's what's so important, that middle box. The data generation should be generated quickly, but as you've heard over time, especially with these new therapies, it takes time for the data to mature. It takes time for patients to relapse. We don't want patients to relapse, and then we want these insights to get to the clinic. So you want that middle box to represent speed. So whether it's helping companies prioritize, the Dream Factory prioritize which compounds to bring forward, whether it's filtering out the noise that Larry and Clifton mentioned, whether it's speeding new diagnostics that may do away with bone marrow aspirates. All of that is really important, and I'm really glad that the panel was able to make that the value of that middle box so clear.